A warm welcome to all. Today we see about the topic on viral biopesticides in the toxicology of insecticides course presented by myself. Next move on to the topic biopesticides. In the term itself, it derives the meaning that is from one living organism we control the other living organisms. That is the definition from the US EPA, that is United States Environment Protection Agency, derived from natural materials such as animals, plants, microorganisms, and certain minerals. The biopesticides are extracted. At currently, there are 299 active ingredients are registered and 1401 biopesticide products are registered. Next, why biopesticide? We must think of why we need the biopesticide usage. Because by using the synthetic pesticides, the pest resistance develop easily because they concentrate only on single mode of action. But in biopesticides, multiple mode of actions are happening that reduce the developing resistance of pest. Next, our topic viral biopesticide. In general, the virus meaning Latin word poison. Next, there are virus capable to enter into the susceptible cell that takes part of the cell's metabolic system and makes the cells to die. The nucleic acid of the viruses, which takes part of the cell with a protein case known as capsid. That is the host cell infection process happening here. Next, entomogenous viruses. What do you mean by entomogenous viruses? Means the viruses which has the genes to attack the insects is known as entomogenous viruses. It may be divided into two types, that is inclusion viruses or occlusion viruses. In occlusion or inclusion viruses, there are two types, polyhedral viruses, granular viruses. The polyhedral viruses again divided into NPV, that is nucleopolyhedrosis viruses, and cytoplasm polyhedrosis viruses. Next, non-inclusion viruses are non-occluded viruses. According to the Insect Commission on Nomenclature of Viruses, 12 families and one unclassified group of viruses are pathogenic to insects. It was reported by Miller in 1998. Next, DNA viruses and RNA viruses. That is, the genetic material of the viruses is composed of DNA. Another one is genetic material of the viruses is constructed by using RNA. The DNA viruses are includes the baculoviruses, ascoviruses, iridoviruses, parvoviruses, polynaviruses, and poxviruses. In RNA viruses, it includes reoviruses, nodaviruses, picorno-like viruses, and tetraviruses. Next, what do you mean by occluded viruses? The occluded viruses are the mature virus particle that are embedded with a particular protein matrix, forming paracrystalline bodies, occlusion bodies. That is, the occlusion bodies are susceptible to UV inactivation, that is, incapable to inactivate by the ultraviolet rays. It makes the occluded bodies of the virus to long period persistence in the environment, especially in the soil, to attack other species. Next, mode of action for microbial biopesticides. There are three mode of action for microbial biopesticides: biological or ecological, physical means chemical or biochemical. In biological or ecological means predation or competition, that is naturally occurs. That is, one predation means that one living organism attack the other organism, make them as prey. Competition means it creates a competitive against for food source. Physical behavior means creating barriers or occupying the space of another organism. Chemical or biochemical means it disturbs the biochemical, genetic, or structural function in the targeted pest. That is, alter the organelle structure, function, and their biochemical properties. The family Baculoviridae. In the family Baculoviridae, there are 600 viruses are reported. safe to non-target organisms. Baculoviruses. Baculo meaning rod-shaped, that is baculin in Latin word sticky in nature. You may able to stick on any material easily. The diameters are 40 to 50 nanometer and the length will be 200 to 400 nanometer. It have the double stranded DNA structures as their genome. 37 core genes conserved in all baculoviruses genomes. That is, among the baculoviruses, 37 core genes are conserved for the genome study purpose. Host range are restricted to only insects. It doesn't affect the mammals like human beings. Very safe for insect pest control. Infect orally and occlusion derived variants infect the midcut epithelial cells and dissolute occluded bodies in the alkaline midcut. What do you mean by occlusion derived variants? Seen in next slide. There are two variant phenotypes found in the baculoviruses family. One is occlusion derived variants and another is budded variants. The main difference between the ODV is for horizontal transmission, that is, it transmits from between the host, one host to another host. But in the budded variants, the systemic spread within the host. Within the host, the spread will be systemic from one organ to another. Next, found occluded with proteinaceous crystals. The occluded viruses are found with the proteinaceous crystals known as polyhedra. They may replicate rapidly, makes the cell extension, and to destruct the tissue in host cell virulent mortality genes, capable to cause epizootics. That is, epizootics means easily attack of the insect diseases. Next, the photographs for occlusion body, occlusion derived virus, and budded virus. The difference between the occlusion derived virus and the budded virus are small bud-like structure arrive from cap, the apical cap structure, well surrounded by a bud-like structure in budded virus. Next, the infection cycle of baculoviruses. There are three phases: early phase, late phase, very late phase. In the early phase, the genes are transcribed by the RNA polymerase. Up to 5 to 6 hours post-infection, it happens. Next, in late phase, between 5 and 18 hours post-infection, the genes which are transcribed, which encode the structural proteins, the budding of the nucleocapsid. In very late phase, it happens around 20 hours after the post-infection. That is, the transcription genes are encoded with the structural proteins. It's further the transcription of occlusion specific genes, that is polyhedron gene, in the 10th segment. Here the gene promoters are strong, continue to overexpress themselves. Now the 50 to 75% of the infected cell protein is polyhedrin. Next, the mode of action of baculoviruses. Baculoviruses, that is the host insect larva ingested the baculoviruses. It reaches the alkaline environment of the midgut. In midgut, we know that the peritrophic membrane, chitin and glycoprotein layer protects the midgut. The peritrophic membrane protects the epithelial cells from abrasive food particles and pathogens without abrasion. Here, the viral enhancing factors, that is, the baculoviruses encode the metalloproteases enzymes. Viral enhancing factors is to increase the oral infectivity of the baculoviruses from 2 to 15 fold at normal levels in occluded bodies or ODV. That is, here in this diagram, when the insect larva, host larva, ingested with the polyhedra with the embedded variants, it ingestion through the mouth and reaches the midgut. There is the dissolution of polyhedra, releasing the variants. It will break the peritrophic membrane, especially the intestinal mucus, and makes the cells, epithelial cells of the peritrophic membrane, to disintegrate themselves. Viral enhancing factors that mainly concentrate on the to degrade the structural integrity of the peritrophic membrane, specifically the intestinal mucins. Viral enhancing factors is effectively for the infection in host to increase. Next, nucleopolyhedroviruses. It comes under the baculoviruses, polyhedroviruses. Polyhedroviruses. It is a nucleocapsid that is it's also rod-shaped, double-stranded DNA, length that is 200 to 400 nanometer, width 36 nanometer. Polyhedrin here inhabited in the nucleus. That is why it's nucleopolyhedroviruses. Polyhedro-occluded bodies, that is 0.2 to 15 micrometer in diameter. There are two types of nucleopolyhedroviruses. One is MNPV, that is multiple nucleopolyhedroviruses. Another one is single nucleopolyhedroviruses, that is only one nucleus surrounded by single nucleus only present separately. Combination of multi-nucleus, that is 3 to 4, are known as multinucleo polyhedroviruses. Example, the Trichoblisia species in many multiple nucleocapsid, that is Autographa californica. Next, mode of action of nucleopolyhedroviruses. Nucleopolyhedroviruses is especially host specific in nature. That is majority of the lepidopteran insects, that is Podoptera NPV, SL NPV, Helicoverpa armigera NPV, HA NPV, or we know generally that will affect the specific host for their concentration. It not wide range host attacking. It's the foremost important point in NPV. It enters through the injection of plant materials and reach the gut via mouth and cuticle. Polyhedrin protein dissolves in the alkaline environment of the gut is as like baculoviruses, that is the alkaline environment of the gut. It reaches, then the epithelial cells of the gut and the replication begins. Here the detailed diagrams, that is the polyhedra are ingested via the mouth, reaches through the foregut to the midgut, that is especially the peritrophic membrane. Here the polyhedrohedrin variants are dissolved in the peritrophic membrane, makes the peritrophic membrane to break and cause the epithelial cells of the midgut to disintegrate themselves and cause disintegration mainly on intestinal mucus. The symptoms of NPV attack, it may be the larva may be discoloration, that is the actual color of the larva may change brown and or yellow. It will feel the stress condition, decomposition, that is liquefaction, the ooze out may be come out from the larvae. Lethargy, it will show the died symptoms. Infected larva hang invertedly from twigs. It's one of the foremost symptoms of for the NPV attack in nature condition. That is, if the Spodoptera is attacked by the NPV, the Spodoptera move towards the top of the plant and climb to the highest point, and that hang downwards towards the geotrophic moment. The symptoms is known as commonly treat of disease or caterpillar wilt. From this caterpillar or larvae, the dispersion occurs, the virus may spread to a maximum range. The NPV effectiveness, that factors are necessary, that is it should be applied when the day temperature is between the 25 to 35 degrees Celsius, slower under cooler conditions. It should be under the cooler condition, so not applicable under 18 degrees Celsius. Larval death may take longer time in cooler condition compared to hot condition. The larvae may take in charge only in the outer region, that is in maize. The larva in silk area means it will easily affected, but inside the cob means it is not able to reach the host and attacking it. And it's in also cotton, while the squares are formed, the target of the host may may be difficult to reach by the NPV. Next, there are two types of biocide techniques are screened for mass culturing of the NPV. One is diet plug assay, another is droplet feeding assay. One is diet plug assay. The insects are fed with artificial or semi-artificial diet that is mixed with viral suspension. If the insects consume leaves means the leaves are dipped with the viral suspension and make the larva to feed them and produce the virus multiplication is this diet plug assay. Next one is droplet feeding assay, that is using the viruses fed in small droplets, that is make the larva to feed upon the droplets of viruses nearby them mouth area. Now we see about the production of Spodoptera litura NPV, that is first of all we should take the third or early fourth instar larvae. By dip method we have to before that we may make the larvae to pre-starved for up to 6 hours for the best suited of virus multiplication, because if we pre-starve the larvae they start feeding the feeding material effectively and voraciously. Now the virus suspension containing that is the 10 to the power of 8 polyhedrin occluded bodies per ml of water is mixed with 0.1% T-pol solution. For sticky nature, T-pol is used. Now the castor leaf is dipped in the virus suspension and shade dry for few minutes and allow the larva to feed on the treated leaves and in the untreated leaves. Now the inoculated larva start showing symptoms and die after 2 or 3 days. Now these larvae are collected in the distilled water. We collected the death larva in the distilled water and grinded it. Now the grinded suspension is allowed for several days at a room temperature. The polyhedron gradually settles at the bottom of the container as a white layer. The supernut containing the larval debris are decanted and polyhedron are suspended in water. Now the clear solution will apply as the polyhedron viruses are settled down at the bottom of the container. Next clear solution will be appear over the top. Now we have to isolate. How we isolate means the supernut fluid in the containing that is are suspended in water. Now purify the sediments by using different centrifugal at 500 rotation per minute for 2 minutes in low speed and at 1000 rotation per minute for 10 minutes in high speed. These are the isolation techniques and purification techniques of NPV. Next, how to determine the concentration. The concentrations are determined by the POBs, that is polyhedral occlusion bodies per ml of the finally prepared solution is used to prepare the field dosage. Field dosage is based on the number of larval equivalent LE, that is 1 LE is equal to 6 into 10 to the power of 9 POBs per acre or number of fully grown diseased larvae per acre. Better we recommend 1 LE larval equivalent in the ratio. Next field application. In this the NPV are prepared on the concentration based on 250 LG for 1 acre. To apply the 250 LG, 1 kg sugar cane, 1 kg cane sugar, that is jaggery, mixed with the T-pol to sticking agent 100 ml and a required amount of water are sprayed with knapsack sprayer. Especially knapsack sprayer is importantly to use to spray to cover the plant pods evenly wherever eggs and young larvae are noticed. Next, counting of POBs. POBs are counted by in laboratory condition by the Newberg chamber or hemocytometer. For this, the squares of the hemocytometer releasing few drops of SLNPV suspension between the cover slip and the marker portion. Wipe out the excess suspension within the tissue or sponges. Observe under the research microscope. Counting is done by including the cells touching middle line on top and left and excludes cells touching middle line on bottom and right. Counting is done by the watch the last point, that is including the cells touching the middle line on top and left, and exclude out the cells touching middle line on bottom and right. In this picture, this is first is Neuber chamber. The Neuber chamber consists of the grid, that is squares. In that, the counting of PBs, we see that at top and left we may considered, but at the bottom and right we not considered as the effective. Next, cytoplasm polyhedrosis viruses. It's also belong to the baculoviruses, polyhedroviruses. That is, it's belong to the genus Cipovirus, family Rheoviridae, but isolated from the Lepidoptera, Diptera, Hymenoptera, Coleoptera, and Neuroptera orders. Large damage to sericulture in Asia. Develop only in cytoplasm. Only in nucleus means nuclear body NPV. Only in cytoplasm means CPV. Of the host medical epithelial cells. Virions occluded simply in polyhedral aggregation bodies. Here only simply SCPV, single cytoplasm polyhedroviruses. Tin double stranded RNA. Here the genetic material is RNA. Protein encoded in the 10th segment of the genetic material. Then their characters: 60 nanometer diameter, and POBs are range from 0.5 to 15 micrometer. It's also confined to midgut and does not spread to other tissues. It's the main point. Infection not always lethal, but shows larval growth reduction. Continuously shed infective polyhedra in feces. Induce the chronic and sublethal disease in the host, that is, reduced fecundity, pupal weight, and the survival of the offspring may be caused by the cytoplasm. Maybe not, it's definitely caused by the CPV attack. Next, culturing of CPV. That is, the low efficiency of spread of viruses infection from cell to cell make difficult to culture. The formation of virion or liposome complexes, liposomes, that is, virosomes or lipids. Efficient initial infection of the Lyme-Artra disper derived LD652 cell line, but for a limited duration. So it may not be effectively used for the mass culturing of the CPV. No clear cytopathic effects observed. No functional virions. It's also reported by the Hill et al. In 1999. Next, the granulosis viruses. That is, it's developed either in the nucleus, cytoplasm, or tracheal matrix, or epithelial cells. It doesn't depend upon the specific site for its multiplication. It's developed wherever it reaches. The virions are occluded singly in small inclusion bodies called capsules. It's also rod-shaped virion, that is, baculoviruses are generally rod-shaped virion, double stranded DNA. Here the genetic material is DNA material. That is, next, the oval occlusion bodies are about 200 to 400 nanometer in length. They enter through the ingestion. How they enter means ingestion only via the mouth. One of the major character is it only affect on the fat body, that is the steroidal hormones. It may cause the changes in the hormones and attack the insect's growth and development. Diseased larvae. How may it seems to be? That is less active, flaccid, fragile, not strength, wilted, prone to rupture in later stages. Death in 6 to 20 days. It takes up to 20 days to kill the larvae. Baculovirus' role in growth regulators. That is, the granulovirus' role in growth regulators. That is, the granulovirus' major organ is fat organs. That is the steroidal. That it is also based on the fat hormone. The granuloviruses encode the enzymes interact with the ecdysone, which triggers the molting in the insects. Enzyme ecdysteroid UDP glucosyl transferase. that is EGT. EGT catalyzes the transfer of glucose from UBP glucose to ecdysone. By the inoculation of the granuloviruses, thus inactivating the EGT hormone and blocking the molting processes. By that, the infected larvae died in the molting stage, leads to more production of virus occlusion bodies. In this, there are two types, that is wild type granuloviruses, recombinant type granuloviruses. In wild type, that is natural, it available, that is the 30% greater production of occluded proteins, not blocking the molting processes. It doesn't block the process of the molting, but the larva make up to die during the process of molting. It is wild type is utilized for the more production purpose, but in recombinant type, by the deletion of the EGT gene in the granuloviruses makes the infected larva to kill faster. The effect will be easy, immediate to the farmers or to the targeted pest. For field condition it is effective, but for the production it is not efficient. Next, come to the chitinase gene. The chitinase gene also found in the baculoviruses, that is infecting the lepidopteran insects, that is the larval. It expressed in the late phase, that is early phase, late phase, very late phase. After the genes are transcribed by the RNA polymerase, the genes are encoded with the protein-like structures. We see in the previous slides that in the late phase, that is 5 to 20 hours post-infection time, this gene may exhibit and begin to infected cells to lyse. Chitinase is essential for the digestion of chitin, that is we know exoskeleton. It's also involved in the process of molting. By this, the granuloviruses importantly concentrate on the fat organs such as hormones, the molting hormones, ecdysteroid and chitinase. Next, cathepsin. It's also expressed in the late phase. It helps to break down of the cellular structure, makes the cell structure to degrade and disintegrate. The integrity of the infected cadaver maximize the occluded body dispersion. However, the cadaver may have the ability to disperse the occluded bodies in a very wide range. In this, the recombinant type, either one or both genes deletion leads to keep the cadaver intact for several days without melting symptoms. Either the chitinase or cathepsin gene, both are expressed in the late phase. If either one or make the both genes to delete means the cadaver of the insects, that is the dead pupa, does not show the melting symptoms. It will survive for it will keep for long time. When we need means it takes to the dispersion level maximum. At last, these are the nature of virus and host. For NPV, they are host specific nature, that is Helicobacter parmigiana, Spodoproliptera, Amsecta albistrica, Spilosoma obliqua, Carcera cephalonica, Plusium peponis, Anthurium militia, Diaserica mendoza. For granuloviruses, Aphelococcus modernalis, Aikia janeta, Chiloinfus cutulus. For CPV, Helicobacter parmigiana, Porpox virus, Amsecta mari. The commercial products available in our market are BioKills, SunBio, that is Helicobacter NPV, Helicap. For especially Helicobacter and Spodoptera, the marketed products are available. The baculovirus infection are studied in the Lepidoptera, that is 455 species, Hymenoptera 31 species, Diphtheria 27 species, Coleoptera 5 species, Neuroptera 2 species, Trichoptera 1 species, Thysanoptera 1 species, that is Thrips, Siphonoptera 1 species, and also the crustaceans, that is aquatic insects. These are the 4 review articles I referred for the presentation of viral biopesticides. One is the biochemistry, next the biopesticides mode of actions, next the baculoviruses characters, at last biopesticides efficacy. Thank you.